and I haven't been participating in the other meetings here. So last time it was about nonlinear models without the mixed part. And if you're interested in that, you have the link that Hadra shared. So today, I try to structure my presentation in just 4 questions that I'll answer today. You know, what options do we have, continuing from last time? What do I mean by mixed models? How do we model the random and error structures in nonlinear mixed models? And how do we assess uncertainty in nonlinear mixed models? And maybe some of you were listening already, but this kind of topic of fitting lines to data is in the news now. This is from the New York Times. And, you know, there's been a lot of comments about trying to predict this COVID-19 dynamics from a cubic fit. How many people are appalled that they're doing something like this. I'm not going to comment on this because I don't know anything about epidemiology, so it doesn't correspond, but maybe after seeing the comic that that Gina sent, you have more appreciation about your options. So last time, what options do we have? Last time I talked about polynomials, about classical, what I call classical nonlinear regression. I also mentioned nonlinear regression for quantiles in cases where that is relevant. And also, I'm going to mention very short generalized additive models, because I think it's important that you know about them. Even you know, that could be a separate presentation, but I'm going to mention them briefly in case this is appropriate for what you want to do. So generalized additive models. Generalized means that we can fit more than just normal responses, and I'm not going to go into that today. That's a topic for a different session. But additive, the additive part means that we have linear predictor involving a sum of smooth functions of covariance. And here we have generalized, meaning that we could fit something other than a normal response. We have a linear predictor, which I'm going to give an example, and we add some smooth functions to account for the nonlinearity. And an example where we have used this generalized additive models is for soil moisture. So maybe some of you have data, but it doesn't really match very easily one of these nonlinear classic functions. And soil moisture is a good example because it's fairly driven by precipitation, but also the dynamics of the physics of the soil. And it goes up and down, you know, very unpredictably. And it would be very hard to model this with some kind of function. That, you know, how do you write a function that captures this? You could use polynomials, but maybe by now you already know that polynomials would be a bad idea, even for the sake of interpolation. An example where we have used generalized additive models for modeling soil moisture was an example where we were looking at the effect of cover crops. And we analyzed the data for many more situations than the one I'm showing you here, but I'm just showing you the example is, you don't have the points of data here, but you can imagine there's a bunch of points that you get from soil moisture sensors. And the lines correspond to the fit of those points. And in this case, we use the GAMs that we use are called splines, and they're called cubic splines in this particular case. And we were more interested in asking the question of, okay, you know, are there statistical differences at this point in time between having a cover crop and not having cover crop in soil moisture? So I just mentioned these as an alternative to the things that we've been showing. And I'm going to give you a very short example next of application of these GAMs. So this is an example where this is probably the minimum amount of data that I would use for using this method. But this is an example where they've grown a weed in the greenhouse, and they applied different amounts of stress. And they wanted to know how that stress affected the plant height, for example. So here you have the treatment, and I know I could have labeled these better, but this 1 means no stress, and 2 means a bit more stress, 3 is even more stress, 4 more stress, and 5 the largest amount of stress that they impose within this specific treatment. And I know some of you, I know the ones that do use R a lot, probably use ggplot, and you can easily get you ggplot to give you some lines like this ones. It's like, ta-da, magic. You know, you get some lines and you can fit the points. But maybe this part is more about taking control of this thing that ggplot would do, these lines that would draw through the lines. So lines that would draw through the points. So the code I'm using is a ggplot, and I'm assuming that some of you at least are familiar with this, and you can use geom_smooth. And in this case, I say standard error false because I didn't want those standard error lines in the previous graph. But it might magically draw lines through points like it did in the previous graph. Now, one of the options you have in ggplot and geom_smooth, it does this automatically for you or automagically for you. But you could say method gam in that case. So what it's doing, what it could be doing, if you do that, it's using library mgcv, which it provides one implementation of GAMs, of generalized additive models. And in this case, what's really nice is that I could ask, this is basically a linear model, but I can ask the height as my response, treatment, the stress imposed on those plants. This is days after transplant because it's conducted in the greenhouse. And the interaction between my treatment and day. And then I simply, this is, I think, the appealing part, is that here I just say S DAT. And this makes a lot of assumptions, but it adds a smoothing term, an additive term, to our linear predictors. So this would give you the kind of fit that you saw in the previous slide. And you could ask questions at this point of what's the difference among treatments at different points in time and day. And it accounts for the nonlinearity of the data. I think that the default in this S function they're called thin plate splines. But again, that would be a topic for a different time. Now I want to show you the relationship between this generalized part that we could fit something other than just height. The linear predictors are these 3 terms that you see here in the middle of the formula. And this F1 function, it's a smoothing function of, in this case, our covariate, its time. So that was, I just wanted to quickly mention GAMs to give you something other options rather than the options we've seen, because they're very flexible. They work well when you have something that's very unpredictable, kind of like soil moisture. And they could even be used in a simple example like the previous one to model the relationship between height, plant height, and time. And today, now I'm going to talk about this mix part that I mentioned last time. So last time we talked about the growth of orange trees and how you might have a relationship between circumference and age in days. But what I didn't mention last time, but I hinted at the end, was that these are actually different trees that we're looking at. They're individual trees that have this relationship between circumference and days. And last time I ignored this structure on purpose to keep it simple. Now at this time, it's very common that you have different trees, and for simplicity at the beginning, for this example, I'm going to show you a linear relationship. So we could have something like this, where we have different trees, and for this range of data, we could fit a simple regression to each tree. And you know, at this point, you might have different questions, and in terms of fitting the data, you can think of it, okay, if we have a relationship like the previous one, do we just fit one single regression line to all the data and forget about the structure, forget that there are different trees, forget that the variance is increasing as time increases. Like, of course, you can already see all the downsides of just fitting one single line to all of the data. Or we could also fit a regression line for each individual tree, like I showed in the previous slide. And there are downsides to that too, because now we have a model that is over-parameterized. We're choosing to use too many parameters to describe something that's fairly simple. So in terms of what these models look like, the model for a simple linear regression, we have J indicates the different observations. We have beta 0, beta 1 are the parameters in the model. And we have one single estimate for beta 0, one single estimate for beta 1. We have our covariate, which could be time in this case, and we have our errors. Now I'm using the subscript I to denote the group here. So each individual tree would be a different value of I, and different value of J would be a different point in time here. So we could have a separate beta for each individual tree, and a separate beta 1, beta 0 for each individual tree, and a separate slope for each individual tree. But how do we know that? Do we need to have a separate intercept for each individual tree? Do we need a different slope for each individual tree? So a mixed model, one way of thinking about mixed models is this formulation, which is also, this type of formulation is also called a random coefficients model. And this would be a random effect. You can think of each individual, we have a single intercept and a single slope, and I call them fixed here, single fixed intercept, single fixed slope for all the trees. And you have a random deviate or a random effect on the intercept that depends on I, so meaning that it depends on the tree, and a random effect that depends on the tree for the slope as well. Now we can model, the difference here is that we can model these the same way that we model errors. So we would say that the effect on the intercept, it's a random effect that is normally distributed with mean 0 and some standard deviation. And a B1 that depends on tree, again, it's a random effect that depends on the tree, can have a normal distribution with 0 mean and some standard deviation. And here you can see that we could, this mixed model could end up being a lot more efficient in terms of how we model the data than the one that has a single, an individual intercept and slope for each individual tree. Because we could test for the effect of this variance or this standard deviation. And if this standard deviation appears to be non-significant or close to 0, we could eliminate this from the model and we could have a single intercept for all the trees. And then we make our model simpler. So again, this is the example. And the idea of the mixed model as well is that, all these trees look very similar, right? It looks like they have very similar intercepts and very similar slopes. So it doesn't really make a huge amount of sense to have a separate intercept and a separate slope for each individual tree. So the mixed model allows us to capture this. So when I fit a mixed model to this data, it looks like this. And this graph shows the blue line is the fixed part, which is that beta 0 and beta 1, but it's common to all the trees. The pink line is the individual deviation that you get to when you look at an individual tree, right? So this is how this tree deviates from the average. And you see that we capture the average response and also the tree level response visually, right? When we look at the output from R, we see that we get our intercept. This is beta 0. This is the estimate for beta 0. This is the estimate for beta 1 for the slope between age and the growth. And look at the standard deviations. Those things that I've shown you before they're estimated here. This is the standard deviation for the random effect at the tree level, on the intercept, and the tree level on the slope. We see that the standard deviation for the intercept is very, very small. So that means we could eliminate this term from the model, and we don't need separate intercepts for each individual tree. So this would simplify the way that we're modeling this data. Now, of course, last time I showed you that the logistic function is a better choice for this type of data. And I fitted that logistic function considering it as a nonlinear mixed effect model. And this is what it would look like, okay? So now that you understand or I hope I explained more or less reasonably well, the random effects model, we can think about, a nonlinear mixed model is the same thing as before, but now we just have a nonlinear relationship among the parameters. So for the logistic model that I explained before, we have the asymptote, and we have some random deviation that is due to tree. And for the x midpoint or the inflection point, we have some random deviation due to tree. And for the scale that controls the spread of that function, we have some random deviation beta 3 due to tree. Now, if we put all these b's into a vector that depend on tree, we assume that they're normally distributed, but by now I'm generalizing this a little bit more, and there's a covariance structure that is a covariance structure of the random effects. Now we have sigma 1, 1, sigma 2, 2, and sigma 3, 3. These are, in this case, the variances for the random effect associated with the asymptote, the random effect associated with the inflection point, and the random effect associated with the scale. And then we have off-diagonal terms that are covariances. So now I'm going to switch to a different example because with the orange tree example, we sort of exhausted what we could do. We had different trees, we fitted a logistic function, and we fitted a nonlinear mixed effects model, which accounted for the fact that we have different individual trees. So now, because I'm an agronomist, I chose a soybean example. And I thought about choosing a different kind of example, but the appeal of this is that this data set is distributed with the NLME package. So it makes it really easy for you to reproduce what I do. That's what I chose it. In this case, we have soybean plants, and they measure the weight of the leaves in the soybean plants. And we have time on the x-axis. And this is a whole bunch of different plots. But we don't only have different plots, we have two different varieties that we're looking at. But this example is going to give us the opportunity to look at a bit more complexity than what we had before with the orange tree example. So one of the things I do as a preliminary step to fitting a nonlinear mixed model is I fit an individual nonlinear regression to each one of my plots, in this case. And each individual plot is the experimental unit where you actually take the repeated measurements of weight or height or anything else. So here you see the asymptote, the variation in the asymptote of those soybean plants, the inflection point, the variation in the inflection point, and the variation in the scale. There's one plot here. There's one plot here that couldn't fit a line to that point because the algorithm didn't converge, but that's not a problem. And there's one here that you would call this a false convergence. It looked like converged, but the confidence intervals are huge. So that means there was a problem with that plot. But this is not a big deal when fitting this kind of models. What I want to show you is how do we fit this type of model. And I think, for some things, honestly, there's other software, there's many options for fitting this type of models, but what I actually like about R comparing it to other software is how easy it is to do to fit this type of model and the kind of tools that you have for exploring it. So in this case, this NLS list is a function that cycles through all those plots that I've shown you in the previous figure. And this self-starter means that we do not need to provide starting values. We have time as our predictor for our X variable. We have the asymptote, the X midpoint, and the scale. And then we have the soybean data that we're looking at. This plot intervals fitted the plot that you saw before, and I layout is just 3x1 because I made it into a 3x1 structure to fit well in the previous slide. And the function NLME just takes this object that fitted an individual logistic curve to each one of our plots, and it fits a nonlinear mixed effects model. So the fact that you can do it this easily, I think gives you time, or gives you the opportunity to spend more time kind of thinking about how to model that structure. So as I mentioned before, so if you run these lines of code, you just fit it in a linear mixed effects model. So if you think about the model that we were fitting before, now we have to think a little bit, and we have to do a few tests to think about how do we model this covariance structure. And this is covariance structure among the random effects in our model. When we look at the output, we get an estimate for the asymptote, an estimate for the inflection point, and an estimate for the scale. And this output that you see here is that covariance structure that I mentioned before. So these are the standard deviations. If you elevate this to the power of 2, you get the variance. And because covariances are very difficult to interpret for almost, I'm guessing, any human being, it's easier to interpret the information in the previous covariance structure in terms of correlations. So we see that we get kind of high correlations. Just a second. We have high correlations here among our random effects. And this one is a little bit concerning because correlation of this type so high tells me that maybe we're overfitting this and maybe we need to eliminate this. But we're going to do a few tests to find out what's really going on here. But basically, I want to point out that these numbers that you see here are the estimates of that covariance structure that we saw in the previous slide. So I want to answer this question now. How do we model the random and error or residual structures in this nonlinear mixed effects model? So we can visually see the relationship among the random effects in our model. And I should say here that this correlation here that is between the inflection point and the scale seems to be a little bit too high and seems to be a little bit suspect. We'll see how we can kind of resolve this. But you might think, correlations in this case are good because if parameters are correlated, means that there's less, if they're completely independent, there's going to be more uncertainty in our predictions. If they're correlated, they allow us to reduce the amount of uncertainty when we do simulations from these predictions. So going back for a second to what a linear mixed model looks like, we have observations here, and the eye here represents different groups, just like it represented different trees before. And what's important to think about in a mixed model is that we have some part that is fixed, or we can call it deterministic, some part that is random, that we do need to put some effort into modeling, and some part that is a residual. Just a second. Sorry about that. So here we have random effects that we assume have some normal distribution and with some covariance structure, and we have some residual terms that also have some structure here that we might want to model. Now, it's very common that to think that this different structures are competing. And the reason why they're competing is, you think about it, is if you have some observations that you collected, and that gives you some amount of variability. So you have like 100 points. Now you have to think that there's a trade-off between how much each of these three components capture in the total variability that you're looking at. If we are able to capture a lot of the variability in Y by modeling X and beta, then there's going to be less variability that is going to be captured by a random term and a residual term. In the same way, if X beta captures a certain amount of variability, and then you're left off with a certain amount of variability that you have to distribute between your random term and your error term, then there's going to be a trade-off. You can't just model a very complex random term and also a very complex residual term. You either model a lot of complexity here, or some complexity here and some complexity here. So there's a trade-off between modeling these two covariance matrices. In many models that we typically work with, is fairly common that if you put a certain complexity here in the covariance structure of the random terms, you're going to end up with just residual variance that is relatively simple. And this is, by the way, an identity matrix. So meaning maybe you just have one parameter for the residual if you capture a lot of the variation in the random term. Now, the jump from linear models to nonlinear models is not straightforward, and there's more than one way to do this. But the example I'm giving you here is just where you have some function that relates those sort of not fully deterministic, but the coefficients in that nonlinear formula and some covariates. In this example that I'm giving you here, this function was a logistic function that relates those coefficients to those covariates. Now notice now that the random terms enter these coefficients linearly, meaning that we have some fixed part or deterministic part within those coefficients. And remember my random coefficients model, there can be different levels of random terms that are added here to determine our coefficients of our formula. Now, in this case, I have many terms because I'm giving you an example where maybe in ecology, and I think in agronomy, in other disciplines, sometimes we might have a region, we might have different sites within that region, we might have different, different transects within those sites, and we might have different observations within those transects or different sites. So in some models that I fitted, maybe you have data from different states and different climate districts within those states, different sites within those climate districts, and different observations within those climate districts. So you can model different sort of nested levels of complexity here if you want to. So you can make this model fairly complex. One of the things I want to point out too is that this F here in nonlinear mixed models hopefully capture a lot of the variability in Y. Otherwise, you're probably want to be fitting a different kind of model, but the hope is that this F functions do capture a lot of the structure in your data. Now, I wanted to show you the R code here of how you would incorporate the effect of variety, which in the case of soybean example, we have two varieties. And what I do is I extract the fixed effects from my previous fit of the soybean nonlinear mixed model. And I say, well, this fixed term has an asymptote and inflection point and a scale, and this tilde says that it depends on variety. When you're making the model a little bit more complex now, you do need to provide starting values, and that's why I extract the fixed effects. And you see that I take the first one, which corresponds to the asymptote. I take the second one, corresponds to the x midpoint, and take the third one that corresponds to the scale. These zeros represents the variety effect. So if you have had a class of ANOVA where you see analysis of variance, you probably remember that there's some parameter that can be assigned to one of our groups. And this other parameter, placeholder 0 here, is a deviation from, let's say, group A to group B. So this 0 represents that deviation from one group to the next one. And this is sort of like, sometimes you state, my null hypothesis is that the treatment effect is 0. So there's no difference between the two varieties. Well, these are the zeros that you put in here. But these are just starting values because we don't know maybe a priori what is the difference between variety A and variety B in terms of how they affect the asymptote, the x midpoint, and the scale. When I fit this model, I get a test and it tells me that, well, maybe there's no effect on the variety on the asymptote. This is just a test for the fixed part of that coefficient, which if you have a reasonable nonlinear model, it should be significant because it just means that that coefficient or parameter should be in the model. And this is the effect of variety on the asymptote, the effect of variety on the x midpoint or the inflection point, and the effect of variety on the scale. We also looking at the random effects. Here we see that they look similar to what they looked like before. But there's a high degree of correlation among our random effects. And this, again, is somewhat suspect. So I thought, well, can I fit a model where I eliminate the correlation between the inflection point and the scale? And I'm also going to remove this column and this row, but not this scale. So I did that. And this is how I do it. And should I eliminate the correlations between the scale and the asymptote and the inflection point? And the way you do this, you can define a blocked, it's called blocked correlation structure. And this is the correlation between the x midpoint and the asymptote. And I leave the scale as a separate component. If you look at the AIC and the BIC, you see that they were actually lower with my original model. So it's telling me at this point, it seems like I shouldn't really eliminate those. But they're troublesome. Like having correlations that are that high are a little bit suspect. So what's going on here? It must be something else going on. So let's look at our residuals. Well, it turns out that our residuals show this pattern, this increasing pattern. So this shows that as the fitted values increase, the residuals, the spread of the residuals is increasing. So while we were trying to play around with modeling this covariance structure, maybe it means that we need to do something about the covariance structure of the residuals. So what I did is I actually fitted a model that says, well, yes, as the fitted values increase, the variance should be increasing. When I fitted that model, I looked at my results again, and now the correlations are -1 and 1. Well, now this is a strong signal that now my model is over-parameterized and I should eliminate these correlations. Now, when I did that, after I did that, I looked around the model a little bit more and I fitted models of different complexity. And I'm going to try to explain this. So these were my original models where I did not model the covariance structure of the residuals, FM1, fit model 1, and fit model 2. Once I incorporated the fact that the variance is increasing, as the time increases, then the AIC and BSC dropped almost in half, right? And then I looked at other models where I actually eliminate the covariance for the random effect on the scale. Now, what I ended up with is graphically, the covariance structure of the random effects and the covariance structure of the residuals look like this now. I have a single term for the asymptote and a single term for the inflection point. And the other ones end up being zeros. Like I don't need to have those in my model. It's too many parameters, too complex for the kind of data that I have. The covariance structure of the residuals now looks like I have a specific model for this, but we cannot assume an homogeneous variance, the same variance for all of my residuals. So now the variance increases as the fitted values increase. So, and you can see here that the AIC and the BIC were lower as I made some changes here and I reduced the complexity of these two covariance matrices. So modeling the error structure and the random structure can be fairly complicated. Here I'm just showing you some examples of ways in which you can do it. And it takes a little bit of experience and knowledge about the kind of data you're fitting and so on, but I'm showing you some. I think one of the main messages is the trade-off between the two covariance structures. How do we assess uncertainty in this type of models? And this is something that you can look at different things, but for this purpose, I provide in my package in NLRAA package, Nonlinear Regression Agricultural Applications, this boot NLME function, which can do bootstrapping of our models. So what I do here is I expand a grid. This creates a data frame with variety. I have two varieties here. The varieties are called F and P. And I create a vector that goes from 10 to 90, which are the days, the time, and days. Here I predict create a function that I get predicted values. And here level 0, what level 0 means here is I'm looking at the level of variety, not at the level of plot. We could change this level to ask different kinds of questions. But basically, I'm looking at the fixed level of variety for my predictions. This function, this system times it because I want to see how long it takes, and it takes like 2 or 3 minutes to run. And I take the fitted model 5, the best one I found through the previous process. I provided this function, which takes the predicted values at this level, and I run the model 1,000 times. And this parallel multi-core means that I used 4 processors on my computer. And then I do this to get the quantiles of my bootstrap simulations. So that's the R code, and this is one way of assessing the uncertainty in nonlinear mixed effects model, because NLME doesn't give you any tools to do this by default. You have to do it somehow by yourself, kind of in your own way. So what I'm plotting here is the difference between variety F and variety P for this data set, and the bands are the confidence bands around our fixed terms, or the fixed lines for these two varieties. The ANOVA table here tells us that the variety has a significant effect on the asymptote, but not so much on the inflection point or on the spread of this function, or the x midpoint on the scale of this function. But this is hopefully an acceptable graph of our nonlinear function that we fitted in this context of a mixed model. But when I look at this, when I made the simulations, I thought, it looks like our logistic function doesn't capture the data quite as well, because the points go up and they seem to go down a little bit in our last time where we fitted the model. So I fitted a different function that I presented last time, which is the beta growth function, which allows, the logistic function doesn't allow for a drop at higher values. So I fitted the beta growth function, which allows for a slight drop at the end, as our last time point seemed to have lower values for leaf weight, which is presumably when the plant started to have senescence and translocation to the grain portion of the plant. And the residuals, these are standardized residuals, meaning that they're divided by the estimated variance across our fitted values. They look fine. They're evenly distributed between -2 and 2, which, you know, they're normally distributed. That's what you would expect, and just very few outliers beyond that. So in summary, the kind of questions that I try to answer today were, you know, what options do we have to fit this nonlinear relationships between some X and some Y? And these are not all of them. So I've shown you polynomials, classical nonlinear regression, and GAMs, which are very flexible, which I just touched on today. Could be a topic for a different presentation. I try to explain what I mean by mixed models. And mixed models sometimes are called multi-level models because they account for population levels or fixed levels or some average levels, and they also account for individual levels. So when you have individual trees or individual sites that you want to model, or individual, or they're called subjects sometimes, or experimental units. So you can both look at both of things. You don't have to fit a model either to all of your data at once or to individual experimental units. So how do we model the random and error structures? And basically, I wanted to show you that there's a trade-off. There's competition between how well you model the fixed part, the random part, and the residual structure of your model. Like, you can't have too much complexity and all of these at once. You have to split it in these three levels. And how do we assess uncertainty? You can look at confidence intervals, which you could also derive using Bootstrap, because sometimes the confidence intervals in this type of models are not really reliable based on the normal approximation. You can use simulation, which I didn't show, but I do have in my package, but some examples. And you can use bootstrapping to try to account for that uncertainty in any part of the model, really. So with that, that's all I have for today. But Gina sent us that comic that was really interesting. And you want to look at all the jokes, right, and comments from this this author. But I thought I like this one, that the sudden increase in trends and what people are Googling during this time. And one of them is flour, which I admit I'm one of those people who is making bread at home with this time. But with that, that's all I have for today. And I'll take any questions now.
Thanks, Fernando. If anyone has any questions, feel free to speak up, or you can use the raise your hand function in Zoom if you want. But I think there's a few enough of us that you can just jump in.
Yeah, or if you want to send a message, if you want to use the chat window to send me a message, that works too.
No, this is Philip. Just a quick comment. Nice, very nice job.
Thank you, Philip.
They're looking at the code. Probably get the idea that it's a little bit more complicated to specify, especially a nonlinear mixed effect model, which is absolutely true. And so they're fantastic. They're powerful. But sometimes you end up tearing your hair out trying to get one to fit.
Yeah, you know, Philip, that's in part, that's part of the reason why I wrote this bootstrapping routine, that now I have experience with these models and I can get something to fit. And with the bootstrap, I can look a little bit more in detail about different parts of the model, which, you know, maybe sometimes it's amazing, but sometimes one or two weird data points are causing the lack of convergence. And it's hard to know. But you know, I've used NL mixed in SAS many years ago, and one of the things that was difficult for in the example in SAS was that you do need to provide starting values, and if you don't, it assumes that the starting value is 1. So one of the things I like here is that you can provide starting values in a very intelligent way, you know, which helps, I think, make these models fit, which are they're very, very sensitive to the choice of starting values. Once you're trying to fit, you know, a nonlinear mixed model, it becomes very tricky to get it to converge.
Mm-hmm.
Fernando, I have a question.
Yeah, Gina.
So I'm new at this. And what I have been doing previously is there is a function called like confint, I think.
Yeah.
And that'll give you a range for your for the random effects.
You can get intervals for the random effects, yes.
Yeah, yeah.
Or confint is a sort of generic function in R that people have developed it for a lot of different packages, and you can get confidence intervals for different quantities. But that works, you know, for different types of models and model packages in R.
Yeah, that's why. Are those reliable
Yeah.
for this kind of model? Because I know sometimes like there's functions that you shouldn't use in certain cases, and I always.
Yeah, I think part of the reason I think for using Bootstrap is to try to sort of double check sometimes confidence intervals in models that models like this one and other models sometimes, you know, make assumptions. This model assumes that you can approximate some of these things by normal distributions, and sometimes the assumptions of the model are not met. And
I see.
when you use Bootstrap, you get, you know, kind of an empirical distribution of some of the quantities that the model spits out, and you can compare those to the confidence intervals that do assume a normal distribution.
Okay, that's helpful. Thank you.
And that's true even in just nonlinear regression in general, even without considering the mixed model. The distribution of the distribution of the parameters is actually not normal, and when you look at it, it doesn't really match what the theory says, because the assumptions are not really met.
That makes sense. Well, thank you.
Don't mind my chiming in to add a bit of perspective. Remember, in a nonlinear model, you're freed from the assumption that the parameter enters linearly, which means if you had a model where pH was one of the variables, you could re-express that nonlinear model in terms of the effect on the hydrogen ion concentration, or a parameter instead of the asymptote, you could use the log of the asymptote, and you have exactly the same model. Of course, then when you go to get confidence intervals using confint, you will get different confidence intervals. They don't transform. And the bootstrapping avoids that. You will get the same intervals bootstrapping no matter how you choose to parameterize the parameters. Because it is the issue of confint assumes that the sampling distribution is normal, and the bootstrap gives you an idea of what it is empirically, not by assumption.
Cool. It's 101, so I want to make sure everyone can get on with their day if they were planning on it. Thank you, Fernando. Based on this, like, I would like to schedule a talk about GAMs at some point, because I always struggle with soil moisture data, and I had never even considered using a GAM, so I might hit you up at some point next month.
Okay, maybe for the fall. Are we done this semester?
We'll give you a little break.
So, but thank you so much for taking both of these great presentations. So,
yeah, definitely. Thank you.
Yeah, I think so. Halder and I, so Dr. Dixon is going to present in 2 weeks about some stuff he went to, or he learned at a conference about trying to be more reproducible, I think, with our R coding. That'll be May 22nd. So.
And I think we'll try to do it, have it every other week through the summer if there are people that want to present. Especially, I think a lot of people might be, if we don't have as much fieldwork as normal, working on manuscripts and such, and this could be a good opportunity for students to kind of get feedback on an analysis for a manuscript or chapter, something like that. So if any of you want to present something, email Gina and I, and we will get you on the calendar. If no one's up for it, or if no one volunteers, we won't have something that week. We will, Philip did something about the statistical ecology conference in June, and I think we'll send out some more announcements about that come June as well, but just to get people excited about certain talks. I'm excited about it. Cool.
Yeah, the program for ISEC has not yet been made. I'm on the organizing committee, so I will share that program as soon as we end up getting it.
Fantastic.
But they're trying to have a virtual conference for people all around the globe, which means some of the time zones don't work very well.
Yeah. And I also recorded this session, so I will send that out hopefully today, if not next week.
Great. Thank you, everyone. Thanks.
Thanks, everyone.
Thank you, Halder, Fernando,
Thank
Gina,
you, everyone.